everybody, my name is Reagan Brummett, and for my interactive case study 6 on Oncovin, I'm going to be discussing cancer treatment from a different perspective. Not DNA mutations, but the physical structure that allows cell division. So when we think of chemotherapy, we think of harming genetic material, but certain medications target cell division scaffolding also. So CHOP therapy includes vincristine, or we're going to say Oncovin in this presentation. So Oncovin attacks tubulin, which creates microtubules and not chromosomes. So I'm going to be talking about tubulin promoting mitosis and how Oncovin destroys it so effectively that quickly dividing cells are not going to be able to survive. I'm going to be starting my presentation now. Thank you. Starting off with my slide 1 where Oncovin correlates to CHOP. So the CHOP regimen for certain blood malignancies such as non-Hodgkin lymphoma is described in my treatment table to the right of this slide. So when used together, the 4 drugs, cyclophosphamide, doxorubicin, vincristine, and prednisone, have been shown to dramatically enhance clinical results, which I'm referencing this from Koffner et al. 2002. Vincristine actually has a potent effect, even though it is administered at a rate of only 1.4 milligrams per square meter. By interfering it with the tubulin, the structural protein needed to construct the mitotic spindle disrupts this physical process of cell division rather than causing damage to the DNA, as some other medications do. CHOP works successfully because each medicine inhibits cancer development differently. Now on to my next slide where I will be discussing the building blocks of the microtubules. So my image focuses on the tubulin's molecular structure. Alpha and beta tubulin heterodimers are shown by the red and gold pair. These dimers build long cylindrical structures known as microtubules by repeating and stacking these lines like bricks. So the image's cross-section demonstrates how the dimers align to form a hollow ring. This tubular structure functions as the internal scaffolding of the cell, supporting intracellular mobility, organization, and shape maintenance. And this is all according to Molecular Biology of the Cell, which is the textbook. So later on, the identical structure serves as the backbone of the cell division-related mitotic spindle. Now that I've discussed the building blocks of the microtubules, I'm going to be discussing the function of them in metaphase. So microtubules have been reorganized to produce the cell's pole-extending mitotic spindle. All duplicate chromosomes align along a metaphase plate and attach to spindle fibers in the middle. Before separation, spindle attachment tension confirms chromosomal positioning. This method presents chromosomal laws and ensures genetic inheritance during division, according to Alberts et al. Without correct spindle formation, this cell cannot proceed, and it's unable to occur. This aligns perfectly with my next slide titled How Cell Division is Disrupted by Oncovin. So vincristine's interference with the division process is seen in this figure to the right. The medication attaches itself to the tubulin dimer's beta tubulin component. Tubulin can no longer form a stable microtubule after that contact. And I got this information from Dumontet and Jordan 2010. Chromosomes are able to remain in metaphase because the mitotic spindle, because it never completely develops with the absence of microtubules. So the cell stays frozen until it is ultimately causes apoptosis, which is programmed cell death. Jordan and Wilson, 2004. Cancer cells are particularly susceptible to vincristine because of their fast division and heavy reliance on this exact spindle formation in this image. Now, with being able to understand all of this, we can focus on the causes of Oncovin side effects. So one of the most frequent effects of the vincristine is explained by this neuron-focused image to the right. Microtubules serve as the, let's say, intracellular highways in nerve cells in addition to forming the mitotic spine, while dynein returns materials to the cell body. Kinesin proteins carry cargo toward the axon terminal, Alberts et al. This transport network is inadvertently disrupted when the vincristine destabilizes microtubules. Patients may experience numbness, tingling, or neuropathic pain as a result of the neuron's inability to support nutrients and signaling molecules, Domenta and Jordan. Due to their heavy reliance on microtubule-based transport, neurons are just collateral and are not the focus of this medication, however. Now for my last slide, we're going to talk about the treatment of cancer through the targeting of tubulin. So for this last figure, which really zooms out, it reveals the tubulin has several different drug binding domains. This implies that various chemotherapy classes may target microtubules in many different ways and usable ways. Medications like taxanes maintain pre-existing microtubules and also prevent their disintegration. Vinca alkaloids such as vincristine inhibit polymerization, Jordan and Wilson. These medications have diverse effects, however, but they all prevent cancer cells from completing mitosis. Since many tumors have a weak and altering microtubule dynamics, tubulin has emerged as a crucial therapeutic target and can be used in the treatment of cancer.